Good afternoon, physiology. This is our last week virtual. I'm excited to be back in the lab. I hope you guys are too. It'll be a big change from the way we've been doing things here. And because we got delayed so late in getting started with the in-person labs, we're going to talk next week about how we're going to do those labs. And you're going to need your lab notebook, finally. So any kind of notebook is fine. I'm very flexible about that. And we'll go through how to fill out the lab notebook in lab next week. Okay, so today's lab, you should have a copy of the worksheet, either a hard copy or a digital copy, or at least a piece of paper where you can take notes. It's better if you have a copy of the worksheet so you know the questions that you need to be able to answer based on what's going on. This lab is not going to be in the notebook. That all starts next week. So through the end of this week, we're doing remote labs. Things are going to be very, very different starting next week. But I'm going to go into that next week in lab. You just need this worksheet today. All right, some of you may not have been to campus, possibly ever. So even if you've been to campus before last fall, you're probably not familiar with what we have to do when we come to campus now that we've got these COVID restrictions. So I'm going to walk through a lot of these very, very simply. Before you come to campus, there is an app that you need to download. It's called My VCCCD, and it's available in the App Store, both for Apple products and for non-Apple products. So you need that on your phone, and you will need to upload your COVID vaccine information to the college. We aren't requiring boosters, but we are requiring the vaccine if you come to campus. The day you come to campus, you need to, before you come, open up your app. There's going to be a series of questions. You answer those questions, and it will tell you if you are cleared to come to campus or not. You're going to get that notification right there in the app. Then you go to campus, and there are several check-in locations on campus. The parking lot by the science building has one kind of tucked back in the corner. You go to the check-in location, you show them your phone and say, look, see, it says I can come to campus, and they'll give you a wristband. These wristbands are different colors every day, and they're a way for faculty and staff to look and at a relatively quick glance, tell if you're okay to be on campus or not. All buildings and a lot of the rooms are going to have a QR code posted either outside or inside. We're supposed to be scanning those when we enter a room or a building to help with contact tracing. If anybody in our lab group comes down with COVID, I will make sure that everybody in the lab group knows that we have potentially been exposed. Contact tracing is how the college is doing that, but I will make sure to let you guys know if that becomes something that we need to be worried about. We are requiring masks indoors at all times. We're not specifying what kind of mask. Right now, I did get hold of a couple of the N95 masks. They are seriously uncomfortable. And they get tangled in my hair. I've got this weird curly mat up here, and it gets tangled and pulls on my hair. So what I'm doing is I'm using double masking. I've got a surgical mask, and then I've got a cloth mask over it. If you don't have masks, let me know. I've got plenty of surgical masks. I can get hold of those for you guys, and I can bring those to lab. Your mask has to be over the nose. If it's below your nose, it's not doing anything for anybody. So I know some people, the mask tends to slip down. You can take those ear strips, and I'll show you in class, in lab, if we need to do this. You can twist the straps to kind of make them a little bit shorter. I've seen nurses do that a lot. And that can help hold the mask in place better. Those neck gaiters, you know, it looks almost like a handkerchief that somebody's put over their mouth. Yeah, those don't count. Those do not do much to protect us all against COVID. Surgical masks reduce your risks of the Omicron variant by 65%. No, cloth masks by 65%. Surgical is like 80%. And then the N95s is even more than that. So if you want surgical type masks, let me know. I can get hold of those for you. Okay. Questions on how we're coming to campus? I know it's a little confusing, especially the first time you do it. So you might want to give yourself some extra time.
And sorry, I don't know if you'll know this answer, but do you happen to know if like every class that was scheduled to be in person is coming back, or is it only some?
It is supposed to be every class that was originally scheduled as in person.
Okay. Yeah, I just wasn't sure because I think last week one of my professors mentioned something about lectures not going back, but I think, I don't know. I wasn't sure about that.
There's been a lot of discussion among faculty and administration about what's coming back and when. But the last I heard, everything that's in person is in person starting on Monday next week.
Okay, sounds good. Thank you.
You're welcome. Any other questions on all of this?
Hey there, I have a question. Do you know the situation with parking? Do we need a permit?
We do not need permits this semester.
Ooh, awesome. Okay, because when I was buying my classes, I noticed they weren't available, so I wasn't sure if they were free or if I had to go in person or something like that.
Nope, they're not even checking.
Awesome. Thank you.
You're welcome. Okay. So next week's lab, we're going to be doing dilutions. And so I wanted to kind of introduce you to the idea of pipetting. We're going to be doing it in person next week, but this is a skill that is very important for you to learn. And it's a skill that it takes some repetition for students to get the hang of this. If you've used pipettes before, you're ahead of the game. But most of you guys have probably never handled one of these. So what I'm going to do is I'm going to, oh, it's not going to let me do that. Yes, it is. Hang on. I'm going to copy the YouTube URL into the chat. And I'm going to ask you guys to go and watch that. And then in 5 minutes, we're going to come back to the Zoom meeting and talk about what you've seen and a few things that are really important. Why is this not coming back? A few of the things that are really, really important for us to all remember about pipetting. So go ahead and go have a watch. Okay. You see a couple of people are not back. Oh, here comes one. All right. Hopefully that was useful. Next week, I'm going to demonstrate how we do this in person. I'm going to walk around and make sure everybody understands how to do this. I do want to make a couple of really important comments here. Always use a tip. So your pipette will look something like this, but you see this plastic piece on the end? That is a pipette tip. You always use one of those. And those are used one time and disposed of. And there will be a little beaker or something on the lab bench for you to dispose of them in there. The other thing is never turn the pipette out of range, tune the pipette out of range. So the pipettes are going to have a measurement on them, and they'll say they're a 100 microliter pipette or a 200 microliter pipette. And if you need 205 microliters, you can't use a 200 microliter pipette. You have to go up a size. You can totally break the mechanism if you try and go beyond the top end of the range there. Okay. So like I said, we're going to be doing this a lot next week, but I wanted to introduce it to you so you would have seen it before we actually get pipettes in our hands. The main focus of this lab are the senses and sensory physiology. So homeostasis. You guys have heard this word before, right?
Yes.
Awesome. So one of the ways that homeostasis is regulated is by using specialized cells. These cells are called receptors. And these receptors detect a physiological variable. Actually, sometimes they're called sensors. So they'll detect body temperature, they'll detect salt concentration in various structures, they'll detect pH, and if that variable gets outside the homeostasis range, then these receptors or sensors trigger a response. And that response is to bring things back into range. So an example of this, if the salt concentration in your cerebrospinal fluid, the fluid that bathes your brain and your spinal cord, if the salt concentration gets too high, you'll feel thirsty. And that'll trigger drinking water, which brings things back into homeostasis. If your skeletal muscle length changes suddenly due to an external force, we're gonna see this when we do the reflexes. That can trigger a muscle contraction, which helps to prevent overstretching of the muscle, specifically to the point of tearing. And you probably learned back in elementary school that we have 5 senses. Yeah, no, that's not accurate at all. We have a lot more than that. So you can see here at the bottom, a list of at least, depending on how you determine or you define senses, you've got the 5 that we all learned about: sight, hearing, touch, smell, taste. But there's also itch, pain, balance, proprioception, which is body awareness. I can tell you without looking where my feet are. I know where they are. That's proprioception. Hunger and thirst, time perception. There's some debate about whether people have magnetic field perception, but certainly other animals do. And up at the top is just kind of looking at the same thing: hunger, taste, smell, sight, touch, body awareness, balance, and hearing. Those are the big ones.
I have a question about itch. Is it like the same as pain?
Itch is not the same as pain. It's a different sensor that sends that signal. There's a lot we don't know about why we itch sometimes. It makes sense if you've got something stuck on your skin for it to become itchy so that you notice and you scratch it off. But in our modern times, it seems to be more maladaptive, maladaptive than beneficial. We itch a lot for no reason that we can figure out, or because of allergies. And then we scratch and we, might create wounds on our skin. So it's a very interesting topic, and one that science does not understand all that well.
I have a question.
Yes.
It's just kind of, it's just kind of fun, but so when an amputee thinks they can feel their legs still, would that be proprioception? Or related to that?
That's a good question. It's probably related to proprioception. I don't actually know much about it, however.
Mm-hmm.
So that's an educated guess I'm giving you there.
Okay.
So as we go through the senses, we're going to find several different ways to organize the senses. And the first one we're going to talk about: special versus general senses. And hang on a minute, there's a fire truck going by my house. I don't know if you can hear it. Okay, there's a whole bunch of fire trucks or ambulances or something going by my house. I wonder what's going on. Okay, now I can hear myself think again. All right, so we can categorize all of the senses as either special or general. The special senses are these guys, the ones that we think about, the ones that we learned about when we were kids. And they use a specialized organ. So there's going to be a specific location involved, a specialized organ of some kind involved. So when we say organ, you can think about the eye, but it's really the retina on the back of the eye that we're talking about. Now, calling them 5 senses is deceptive because there are several types of touch receptors. So we think of a lot of different things as touch, but we have different sensors for pressure versus a light touch versus skin damage. So the kind of response we get, the kind of sensation that we feel depends on what receptor has been stimulated. And the general senses, there's no specialized organ. These tend to be, the receptors tend to be in various places in the body. So they're more widely distributed. You could argue that touch should fall under general senses, but we put that in the special senses because probably historical reasons, like when we discovered how that worked. The general senses are often things we are not aware of. I think this is a better way to describe the difference. We are aware of our sight, our hearing, our touch, our smell, our taste. We are not consciously aware of our balance or our proprioception. Do you know what the pH of your blood is right now? Can you think really hard and sense that? No, it's a general sense. There are receptors in the arteries in our throat that send a signal to the brain if the pH gets off, so the brain can respond. When you get to that part of the semester, pH of blood is critically important to maintain homeostasis. If it gets too far out of whack, that's basically death right there. It's also things like glucose levels in our blood, hunger and thirst, and those sorts of things. Another way to categorize the receptors and the senses is by what type of stimulus it responds to. So there are 4 basic types of receptors. Mechanoreceptors, which respond to physical pressure. Thermoreceptors, which respond to temperature. Photoreceptors, which respond to light. And chemoreceptors that respond to chemical stimuli. And you'll notice that there's more than one sense for basically all of these. So mechanoreceptors are touch, but also hearing and equilibrium and our proprioception. And as we'll see, nociception or pain, anytime physical pressure is extreme, we are going to feel it as pain. If we hear very, very loud noises, we're going to sense that as pain. The thermoreceptors, there are different ones for feeling hot versus feeling cold. And that's basically detecting the temperature. It's also pain. Anything that is too hot or too cold, our brain will interpret as pain. Photoreceptors, that's our vision. And that includes both low light vision as well as color vision, which requires more light. And again, it can be painful. If you try looking at the sun, don't do that, please. It will really hurt your eyes. And chemoreceptors are for chemical stimuli, things like smell and taste, but also a lot of the general senses. So your body detects blood sugar levels by detecting chemicals, the sugar in your blood. It also works for things like hunger and thirst. But again, if these receptors are overstimulated, that can cause pain. Why is it that pretty much every kind of sense receptor can trigger a sensation of pain? What is pain telling us?
That something's wrong or out of balance.
That something is wrong, exactly. That you are in danger of injury, or you have received an injury, and you need to respond appropriately. Yeah, exactly. All right, so going to slip over to the whiteboard for a sec. Oops. You've got your little receptor here, and it's going to send information from the receptor to the brain. Oh man, I cannot draw a brain on this. We're going to kind of do a brain. That's kind of brain-like. So it's going to send the information on a specific nerve pathway. Our nerve pathways seldom change. Once we've developed them, they tend to stay the same. We can change them with repeated usage. So you can change nerve pathways by practicing piano. You can learn to play piano without looking at your fingers, for example. These pathways are going to go to a specific location in the brain. So anything that activates I'm going to get right back to that question in the chat. Anything that stimulates the receptor is going to signal that part of the brain, which means that sometimes there's confusion. If you've ever rubbed your eyes really hard and seen flashes of light, that is because you are stimulating the nerves that normally detect light. And so those receptors, those light receptors, are sending a signal to the brain saying, we detect something, and what they normally detect is light. So that's how we sense that. When we think about hot sauce, anybody like spicy food? The spicy food chemical is something called capsaicin. I'm not forgetting the question in the chat. And capsaicin binds to heat receptors, those receptors that detect heat. So we're not tasting the capsaicin, we're feeling it binding to those heat receptors, and therefore we get the sensation of heat. There's no heat there, but those receptors have been triggered. Okay, brain freeze. Woo! Man, you guys are getting me good today. Good questions. Brain freeze happens when your mouth gets too cold. And how it happens is, last I checked, poorly understood, but it's believed to have something to do with your mouth getting cold, causing your nasal passages to get cold. And in your nasal passages, the nerve endings that detect smell get cold, only they get overstimulated with the cold and send that as a signal of pain. To the best of my knowledge, that's kind of our understanding of brain freeze. It's a really cool question. I may have to look and see if anything's been learned about that since last time I heard about it. Okay. So that's how senses are perceived by our brain. There's another characteristic of senses, and that's what's called activity type. So activity type can be what's called tonic or phasic. Tonic refers to receptors that send the signal to the brain as long as there is stimulus. So it's constant signaling to the brain. A lot of pain receptors are this type of receptor. You don't want to be walking on a broken leg and have that receptor quit sending the pain signal to your brain so that you keep walking on your broken leg, because you're going to do more damage. You want that signal to persist. Phasic are receptors that respond to changes in stimulus. So they adapt to the stimulus and you kind of quit noticing it. This is very, very common with smell. Smell is definitely phasic. If you've ever walked past somebody or stood next to somebody and gotten overwhelmed by the smell of perfume or cologne, and you're like, oh my God, how can they stand that? It's because they don't smell it anymore. We get used to it. And so you put on some perfume in the morning and, you know, a couple hours later, you're not smelling the perfume anymore. So you put on a little bit more. A couple hours later, you're not smelling it anymore. So put on a little bit more. And you can see how that can lead to way too much. Same thing with air fresheners. If you've got air fresheners at home, you smell them when it changes and you get used to the smell and you don't notice if you're using too much.
I have a quick question.
Yep.
So for the tonic, so that's like if you get a cut and you could feel the pain, right?
Yeah, you consistently feel the pain. It doesn't stop.
Okay. How about when you know, sometimes like you get a cut and you don't realize it's like you don't feel pain until you look at it. Is that still under like tonic?
What's going on there is probably that, oh man, these are good questions today. I love it. Personally, most of the time when I get a cut and I don't feel pain right away, it's just a slow process for the signal to get to the brain and for the brain to recognize it. It doesn't seem like it should be. It seems like it should be instant, but that's not always the case. I know with paper cuts, you know, I'll accidentally run my hand along the edge of a piece of paper and I'll go, oh shit, I hope I didn't get a paper cut. And I'll look and there it is. And then it hurts. So the signal just hasn't gotten to the brain yet.
Okay, thank you.
You're welcome. Would phasic also be in regards to the weather outside feeling hot and cold? Whoa, partially. Temperature can be phasic, yes. There is also, and this isn't as noticeable here in California, but if you are exposed to warm temperatures day after day after day, your body chemistry changes and different genes become expressed, they become functional to help you deal with hot weather better. So the first day of the summer that's 90 degrees, it feels a lot hotter than if it's been 90 for 2 weeks. This is really obvious if you live somewhere where the seasons change a lot. Like I used to live in Massachusetts, and the beginning of winter, a 40-degree day felt like I was just gonna freeze to death any minute now, even bundled up. And by January, a 40-degree day, I didn't have to wear my heavy coat. I had adapted to it because of these genetic changes in what genes were active. Does psychology influence any of the receptors? Yes. Yes, there can be psychology involved. There's a lot of research right now on using psychology to help people deal with chronic pain. Chronic pain can happen for a lot of different reasons, some of which involve psychological trauma. So yeah, there is a psychological influence on some receptors. I've never heard of psychology influencing things like hearing or vision, but I wouldn't rule it out either.
Like, would hallucinations be an example of that?
Hallucinations are not actually related to the senses.
Okay.
That's all going on inside the brain.
Okay.
Sneezing tends to occur, it's a reflex related to senses. So certain receptors in the nose get stimulated by something that we've got in our nose, like too much pollen or something we're particularly sensitive to or allergic to, and we sneeze to try and get it out of our nose. When you've got a cold, that's a different thing. You're still sneezing to get stuff out of your nose, but that's, you know, a lot of mucus and dead bacteria. Anything else on at this point? I'm glad we're going to be back in person next week because now I'm listening to my neighbor across the street yell at her dog. It's distracting. Okay. So for the lab activity worksheet, we've kind of gone over some of the basics. Not everybody is going to be able to complete all the exercises. That's fine. That's fine. If you have vision problems, you might not be able to successfully do the visual acuity ones. If you have balance problems, you won't be able to do the proprioception ones. Not a big deal. Just let me know if you need help. We can come up with alternatives, or we can come up with videos that you can watch instead. I'm going to go back to the PowerPoint. Oops. Okay. Nope, I'm going to go. I forgot to bring that up. Where is, there we go. On Canvas, on this week's module, there's a whole bunch of links to sites and videos, including all the ones we're going to watch in lecture. And one of them takes you to an image. Really, people, go away. An image like this one over here on the right, called a Snellen chart. It's named after the scientist who came up with this. You've probably all seen this. It measures how well you can see, your visual acuity. Let me go back to the whiteboard so I can write this stuff. And in the US, we talk about vision being 20/20 is perfect vision. The Snellen chart is designed to be measured at 20 feet, so that 20 indicates you should stand 20 feet back. Now, the link that I've got on Canvas is for a version that you can use at 10 feet back, so that you can use it at home. In Europe, instead of saying people have 20/20 vision, they say 6/6 because they stand 6 meters back, which is close to 20 feet. So the first number is how far you stand to see the same as average acuity or typical acuity at the second number. So 20/20, you stand 20 feet back, you can see with, I misspelled acuity again, with an average acuity of what would be average for somebody standing 20 feet back. 20/40, your visual acuity at 20 feet is the same as average at 40 feet. Now, You are going to, at home, use a Snellen chart and measure your vision. Now, this is not going to be accurate. The size of the chart, the quality of your image are going to affect your results. If you have a printer and can print out the chart at the link, not just a random chart, but the one at the link, you can tape it up on your bedroom wall, stand on the opposite side of a bedroom, and you will be approximately 10 feet away. And that's the chart that I've linked to is designed to be read at that distance. If you're doing it digitally on a computer or your cell phone, you would just kind of hold at arm's length. I think it talks about instructions on if you're looking at it on a laptop or a computer monitor, but the size of your monitor matters, the size of the magnification matters. So you might get a number that's just absolutely wrong.
I have a question.
Yes.
Do we do it with glasses or without glasses? Because I use glasses.
Yeah. That is your prerogative.
Okay.
You can decide for yourself if you want to try it with your glasses or not.
All right.
I'm not going to look at it and go, Brazil is such a 20/20 vision, but I see you're wearing glasses all the time. And I just, yeah, no.
Okay.
You're good. The second lab activity is pupillary response. It requires a second person and a flashlight. It can be the flashlight on your phone, that's fine. So you're going to do this at home. And what I've got here are 3 examples of what you might see. The first image, these are normal pupils. You notice that the pupil is the same size in each eye. And this happens even if the light only hits one eye. Your other eye will also set, the pupils will be set at the same size. The middle image is dilated pupils. This happens when it's very dark or if you've been given drops to dilate your eyes. It's to allow more light in. Down at the bottom, notice the pupil on the one eye is much smaller, noticeably smaller than the other one. If you see this while you're doing this, get an appointment with an eye doctor. There are lots of reasons this can happen, ranging from messing up while you're doing this test to something that might be a problem. So you might want to investigate if you see that image. It's not a guarantee there's anything wrong. None of these tests, when you do them on yourself and you don't have a lot of experience, are going to be accurate. All right. So those are going to be on your own. This one, again, I'm going to put the URL in the chat. The video is 1 minute. So I'm just going to give you a couple of minutes to do this. I'm going to ask you to go and watch the video and follow the instructions on the screen. And then let's see what happens.
Did you send the link?
Oh, sorry, it ended up being a direct message to one student. There you go. All right, anybody want to share what you saw?
Genuinely, that was terrifying. I felt like the eyes were like piercing through my soul. That's just my opinion.
Yeah, this is a one of an interesting visual illusion, and it can get kind of creepy. You can do the same thing by staring at yourself in the mirror without blinking. You can get some really weird visuals that way.
I heard that was a thing where if you stare at yourself in the mirror for like 5 or 10 minutes and you just don't have any like outside factors, your brain will start to distort your image. So it's like, oh yeah, like that's where like the whole conspiracy of there's a demon in my mirror comes from or something like that.
Could be, yeah. Or I'm possessed. There, I can see it in the mirror. Yeah. Cool. All right. Now, I want to talk about the next part of the lab activity number 3, which is looking at visual illusions. So the link is here and it's on Canvas. And so what it describes is you go and you look at these visual illusions. I think they ask you to look at 4 different ones and then write about them, because visual illusions can be tricking either your retina or your brain. And in the lab, in the instructions for lab activity 3, it talks about how to tell the difference between those. Basically, if you try it one eye at a time and you get the same result, it's working on the brain and not the retina. Visual illusions are really interesting ways that people trick your eyes or your brain to see something that you're not seeing them. And I think that's a good thing to be aware of because it's easier than ever with like Photoshop. Did anybody see the photo of a guy's found a really large California king snake recently? They kept showing pictures of him holding the snake.
Can't say that I have.
Okay. That is an example of something called forced perspective. Let me,
Like that would be Charlie Chaplin's stunts where he hangs off of the whole clock tower thing, right?
yeah, that's another kind of forced perspective. But I mean, you can see all of these images are designed to make you think that part of the image is closer or further away than it is, and the proportions between those two objects can be really, really different than what they really are. There are a lot of fun illusions out there, but one of the most common ones is snakes. People will hold the snake and they'll often hold it at the end of a stick, and they're holding it towards the camera, and that makes it look bigger than it really is. Those snakes are never as big as they appear to be. All right. Lab activities 4 and 5, you're going to be doing on your own. Activity 4 is hearing. There's some noises that are going to play that are very, very strange. So some people call them creepy. Just be aware of that. Lab activity 5 is looking at your temperature receptors. So when you explain what's going on, make sure you use the ideas of tonic and phasic receptors. So proprioception. Or body awareness. People who have really good proprioception tend to be better at athletic skills than people like me who I'm just no good at that. But you can fool your brain. And we saw visual illusions. There's also an illusion where you can fool your brain about where your arm is. So I'm going to put this link in the chat. The video itself is less than a minute long. What I want you to do is watch the video and then try it. You need to time yourself so you make sure that you lean against the wall or the door frame for 40 seconds. Don't do it any less. The results will not be good. And then come back and let me know how it felt. So, yeah. Okay. If you're back, what'd you think?
I thought it was pretty cool. It was funny.
Good. All right, either people
I
are,
actually notice anything.
you didn't notice anything? Did you do it for the full 40 seconds?
Yeah.
That's too bad.
I remember doing something like this in 8th grade where my teacher would put 2 textbooks on our hands and then for like a full minute and then tell us to like bring our hands down and our hands would like automatically like rise and stuff like that.
Yeah, it's fun to fool your body sometimes. Okay. All right. Now, the last thing we're going to talk about, and I want to talk about it for a couple of minutes first, because I'm pretty sure you guys have not talked about reflexes in lecture yet. So, there go my crappy drawing skills again. When we have a reflex, it is a combination of nerves. See if I can draw these. That act before information gets to the brain. So it's not a very clear drawing. Oh, shoot, I didn't mean to erase it. Okay. So we call these things a reflex arc. And the sensory nerve detects stimulus. The body of the nerve is usually in the spinal cord. And when it detects this stimulus, it sends a signal to the brain and sends a signal back to the body to respond. So the obvious ones that we think about are burning your finger. If you burned your hand, burned your finger, you know that by the time you feel the pain, you've already pulled your hand away. The reflex arc allows faster response to potentially very dangerous stimuli. So if you held your hand on a hot stove, you're going to do a lot more damage than if you pulled your hand back. So the reflex arc allows for that to happen. And there are a whole bunch of muscle reflexes that you can see without doing damage to somebody, without hurting somebody. So the patellar reflex is the one where they hit your knee under the kneecap with a hammer. And that one is designed to, it detects stretching of the tendon, and the reflex which is kicking, is to prevent the tendon from overstretching and getting injured. You have a similar reflex in the biceps and a similar reflex in the Achilles tendon, which is at the back of your heel, not the wide part of your heel, but that skinny part right above the heel at the base of the leg. And those are doing the same thing. They're designed to prevent tendon injuries. And in doctors' offices, it's a great way to test how well your nervous system is working. The one that I wanted to talk about specifically, you're gonna, under activity 7, you're going to be able to go and look at videos of all of these reflexes. The plantar's reflex is an interesting one because it's not 100% consistent. So there's variability here. And there's variability between infants and adults. The reflex is different. The response to the stimulus is different. And so the plantar refers to the bottom of your foot. And so it's a stimulus on the bottom of the foot. In adults, this usually causes the toes to curl down or under. So your toes curl, it causes your foot to twist a little bit. In about 75% of infants, it causes the toes to curl up. So instead of toes curling underneath, they stretch back and stretch out. So spread out from each other. So, you know, if you could see my fingers, they're kind of bent backwards and spread out. But I mentioned it's highly variable. If you go to the doctor's office after taking a 5-mile hike, you're going to have your toes aren't going to curl under. Instead, they're going to curl back. And when they curl back, we call that Babinski's sign. Babinski's sign, toes to curl up. It can also happen if you've just woken up. It can also happen if there's damage to the spinal cord. So there are lots and lots of different reasons it'll happen. And so everybody copied down what I wrote there? So you'll see these links in Canvas. And what I want to do, hang on, I've got to mess with the screen sharing again. I want us to watch this video of that plantar reflex. I don't think this guy says anything. See, the toes barely responded there. Oh, he does say something. It's in German. That's not very much help, is it? Let's try this video instead. So you can kind of see, there go the toes spreading apart. That is what's called the Babinski reflex. And that is normal in infants. I may have to find another video to show you the response in adults. Okay, we don't want to hear you. Okay, dude, just actually do it. Wow. I worked really hard to find a good video, and it turned out it was in German, so that wasn't any good. All right, so I'll see if I can find a better plantar reflex video. It looks like it's gonna take a while. Okay, so that's what we were doing together. You guys can complete the worksheet at home and submit it on Canvas. And I will see everybody in person next week. Unless you got questions, we're good.
Thank you.
You're welcome.
Thank you.
Well, I did happen to have one question, Professor.
Yes, go ahead.
So just in case we had no